Hello, everybody. Today we're going to talk about transcription and a little bit more on the details. And of course, transcription is how we take the message in DNA and change it into kind of a portable form that can leave the nucleus, which is RNA. So let's see. Transcription, three main stages: initiation, elongation, and termination. So initiation is where we start our RNA transcript. Elongation, just making it longer, adding base pairs to it, and then termination, coming to a stop. So start with initiation. Okay, so the three steps of initiation correspond to the three major parts of most genes in eukaryotes and prokaryotes. Okay, so the beginning of the gene has a sequence called the promoter. And this actually, you'll see it a little bit better on the next slide, but this appears in front of the coding sequence or the gene itself, okay? And then this is where transcription is initiated, where it begins, okay? The body of the gene corresponds to where elongation of the RNA strand occurs. We also call this the coding sequence, okay? And then there's a termination signal at the end, and it tells the RNA to stop being made at the end of the gene, and it gets released from the DNA. So let's look at that a little bit more detail. We're going to start with initiation. Okay, transcription binds when we get RNA polymerase bound to the promoter of a gene. So as you know, RNA polymerase is the enzyme that makes a polymer of RNA, okay? RNA polymerase binds to the promoter region, which is right at the beginning of the gene. And the promoter has two main components. One is a site that binds RNA polymerase, and two, a site that one or more regulatory sequences which bind transcription factor or response elements. And these transcription factors and things like that help RNA polymerase to find the promoter and sit down at the right spot, okay? Once we get RNA polymerase to bind to the promoter, the DNA molecule is unwound, pulled apart, right at the beginning of the gene sequence. So let's see what that looks like. So here we have our strand of DNA, and you'll see one strand is called the non-template strand, and the other strand is called the template strand. And so we're interested in the template strand, okay? Because RNA, just like DNA, has to be made in the 5' to 3' direction. It's going to read the template strand, okay? Here's our promoter region. As you see, it's before, here's the starting point of the gene, okay? And then this blue region is our coding sequence, okay? But the promoter is this region here, and there are a couple of essential elements. There's the start point, there's the ta-ta box, and the ta-ta box is very important part of a promoter. It's in every single promoter, and that's where some of the major transcription factors bind. So first, so that's our promoter. The first thing that happens is some transcription factors bind to the DNA, and these transcription factors help RNA polymerase to locate the promoter and locate the beginning of the gene. They help to position the RNA polymerase. Okay. Once they're all bound, RNA polymerase comes and sits down onto the DNA, and it starts to separate the DNA strands. You can see them separating right here, and initiates making of our RNA transcript, okay, by base pairing with the template strand. Okay. So that's initiation, getting everything all ready to go. Elongation basically just makes the RNA strand longer. It continues to add ribonucleotides to the RNA strand, and it reads the template strand, again, by the rules of base pairing. Okay? So polymerase travels along the DNA template strand, starting at the 3' end of a gene and moving towards the 5' end. And again, this is because RNA, like every nucleic acid, needs to be made in a 5' to 3' direction. With the anti-parallel nature of the strands, it therefore has to read the template from 3' to 5'. Okay? Again, the new RNA is complementary to the DNA template, so it follows the rules of base pairing. Okay? And then once the RNA strand has been made, it drifts away from the DNA template strand. It doesn't stay bound to the template strand. It moves away as RNA polymerase continues on. And once the RNA strand leaves, the DNA helix reforms. So the two sides of the DNA ladder come back together. So, and that looks a little bit like this, kind of a zoom in. So here's my RNA polymerase. And you'll see the RNA nucleotides and they're added in based on the rules of base pairing, A with T, U with C. Remember, we've got uracil here. C with G, C with A with T. Next one will be A and T, U and A. Okay? And you can kind of see here is the RNA kind of falling off. Once it's made, it falls off of the DNA and the DNA helix reforms right behind RNA polymerase. And RNA polymerase continues down. Okay, reading this is my template strand, the dark blue strand here, as RNA polymerase is making the RNA from the 5' to 3' direction. Okay, and then that just keeps going. Elongation continues on until it gets to the end of the gene, the end of the coding sequence, and finds the termination signal. Okay, and this is just a sequence in the DNA. It's called the termination sequence. And it basically makes the whole thing fall apart. Okay, the completed RNA strand is released. RNA polymerase lets go and goes and basically just finds another promoter and makes more RNA there. Okay, so this kind of puts everything together. So here we have initiation. So we find our promoter and get our transcription factors, get our RNA polymerase, open up the helix and begin to make my RNA transcript. Elongation continues to make this RNA transcript longer, okay, until it reaches the termination signal. Once it releases the termination signal, the RNA is released, okay. The DNA returns back to its normal configuration, and my RNA polymerase is also released and goes and finds another gene to make. Okay. And basically that's it. It's a pretty simple process. So that's it for transcription. Okay. Now we're going to follow our mRNA transcript to translation, the second half of our central dogma. Okay. So go find that screencast. I'll see you there.